Hello, we are HS 10103 김가은 and 10118 Yunso. Today we will talk about adult and aging brain.
This is today our content. First of all, we will talk about the adult brain and what is aging and how the brain changes and why does the brain age and healthy aging. About the adult brain. Humans lose a lot of gray matter as they grow up. Gray matter here is the excess neurons and synapses in the human brain. In the late 20s, the rate at which this gray matter is removed slows down. At the same time, the brain strengthens its connection with the major nervous system. The main nervous system in the brain is wrapped in an insulated myelin that increases with white matter. At about 40 years of age, white matter in the human brain has the largest amount. The prefrontal cortex is the most important part of the prefrontal cortex that we use to plan, solve problems, and make decisions in adulthood. It is said that adults are more mature than children because the adult brain is connected better for cognitive control than the adolescent brain. Intelligence reaches its peak between the early and mid 25 to 60-year-old adults. But fluid intelligence, which includes the ability to solve problems and identify patterns, is at its peak around the age of 30. In contrast, decision intelligence that deals with knowledge of words and facts increases to about 50 years of age. Therefore, some scientists assume that there is no age at which our cognitive function is at its peak. Resilience is important, both in terms of physical and neurological health, but aging too often increases the risk of injury and disease. One such risk is dementia. Aging is inevitable, but dementia and disabilities can be avoided, so it is better to prevent them in advance. Ironically, contrary to what we know, neuroscientists believe our brains can stay relatively healthy as we get older. The decline in memory and cognitive ability, which was perceived as a normal process of aging, is now recognized as a process of disease in the aging brain. It is natural to lose some neurons as you get older, but extensive and serious neuron loss is not normal aging. The continuous process of aging generally begins in middle age, and study shows that cognition begins to decline as early as 20s and 30s. On the contrary, some studies shows that awareness improves to those in their 50s and 60s before declining. In any case, in order not to get old pathologically, there are methods such as diet and exercise to support cognitive health, and scientists are also studying how to age healthy. Changes in cognitive ability reflected changes in brain structure and chemistry. Imaging techniques have shown that total brain capacity begins to decrease in their 30s and 40s and begins to decrease at a larger rate around the age of 60. However, research on individual brain areas shows that the overall dose loss is not uniform. The frontal cortex, cerebellum, and hippocampus show the greatest loss, but it worsens with age. Several changes in individual neurons can also contribute to reduction in aging brain capacity. Changes in aging are due to a decrease in nerve cells, withdrawal and reduction of dendritic teeth and loss of myelin. In contrast, volume loss in adolescence is mainly driven by synaptic pruning and the death of excess cells. Our cerebral cortex, the wrinkled outer layer of the brain, which includes the body of neuron cells, also becomes thinner as we age. Thinning of the cortex follows a pattern similar to volume loss, and some areas of the brain are most affected than others. Thinning is particularly noticeable in parts of the frontal and temporal lobes. The temporal and frontal lobes are one of the areas with the most distinct decline in both volume and cortical thickness. These are the areas that have taken the longest time to reach maturity. The discovery led to a less-than first-step theory of brain aging, which claims that the last part of the brain to develop worsens first. Among the long-distance fibers in the brain, the first to develop is the projection fiber that connects the cortex and the lower part of the brain with the spinal cord. The fibers that connect diffuse regions within a single hemisphere, the associated fibers, finally reach maturity and show the steepest function decline with age. Subtle changes in cognition are a normal part of the aging process, and memory loss is the most common. But not all kinds of memories are affected by aging. Declarative memory decreases with age, but non-declarative memories are not significantly damaged. Declaratory memory is the memory of learned knowledge that is semantic memory. Non-declarative memory refers to procedural memory, such as remembering how to ride a bicycle or tie a shoe. Working memory refers to the ability to manipulate a piece of information with it in mind. For example, it is to find the phone number and memorize it when you make a call. This also decreases with age. Task memory, an example of fluid intelligence mentioned above, is a set of cognitive technologies that rely on the rapid processing of new information rather than stored knowledge. Other aspects of fluid intelligence, such as processing speed and problem solving, also decrease with age. The ability to focus on a specific stimulus and filter out distractions is called selective interest. For example, when we are in a noisy restaurant, it can be more difficult to focus on what our friends say. In contrast, divided attention refers to the ability to focus on two tasks at the same time. Activities that require this type of split focus, such as having a conversation while driving, also get harder as they get older.
Synaptic changes are selective and subtle, but have a great influence on cognitive decline. As you get older, the number of water bumps and branches decreases, so it gets less complicated, and it also loses its resinous branches, which act as chemical signals. We studied rhesus monkeys and observed that the aging process was aimed at thin spines. The third drone in the second picture is a thin drone. These small, slender protrusions are also high plastic structures, expanding and contracting much faster than larger mushroom spines. This had led scientists to speculate that thin spines can be involved in working memory that requires high levels of synaptic plasticity. Loss of thin dendritic spines can inhibit neuron communication and contribute to cognitive decline. So far, there is a lack of direct evidence of their role in cognitive decline, and further research is needed. Finally, the formation of new neurons also decreases with age. Although it was once believed that neurogenius stops after birth, it adds new neurons from olfactory bulbs and hippocampus to shape vervins. It is not yet clear whether neurogenius significantly affect cognition in the aging human brain, but strategies that promote neurogenius can improve cognitive function. The amount of neurotransmitters and the number of receptors may also decrease with age. Various studies have shown that fear dopamine is is synthesized from the age brain and fear receptors by neurotransmitter. No strange evidence suggests that serotonin levels may decrease with age. Excessive accumulation of abnormal proteins in the brain is known to lead to neurodegenerative disease caused by aging, such as Alzheimer's disease and Parkinson's disease. The accumulation of proteins and other cellular components can also contribute to cell degeneration in a healthy brain. Cells usually break down, recycle, destroy proteins and molecules and are generally efficient but not complete. Over time, damaged molecules accumulate and cells may not function properly, because neurons in the brain are not replaced as often as other cells. Brain cells may become more susceptible to accumulation of damaged molecules. In addition, cellular machines related to the decomposition and recycling processes deteriorate over time, and the efficiency of the waste removal system decreases. Finally, remember that aging brain cells occur in different chains throughout the body. Researchers speculate that deterioration in cardiovascular health, for example, may be one of the many changes in older brain and further drive.
DNA damage accumulated over a lifetime causes aging of the brain and entire body. DNA damage caused by oxidative stress is receiving a lot of attention. Chemical reactions in mitochondria produce harmful byproducts called reactive oxygen, which are highly reactive molecules. If left unattended, it can destroy fat and proteins essential for normal cell function and damage DNA. Our bodies have natural defense mechanisms that neutralize free radicals. Unfortunately, these mechanisms decrease with age, making aging tissue more susceptible to oxidative damage caused by free radicals. Studies of brain cells have shown that damage to mitochondrial DNA accumulates with age. In addition, the brains of people with mild cognitive impairment and Alzheimer's have more signs of oxidative damage than those of healthy people. While the brain's energy needs remain high as aging processes, the brain's energy supply can no longer keep pace. The brain's ability to consume and use glucose decreases and mitochondrial metabolism decreases as well.
Immune dysfunction often occurs with metabolic changes seen in aging. Microglia, a resident immune cell in the brain, performs many important tasks such as protecting against pathogens, cleaning up debris, and helping to maintain the reconstruct synapse. Such as inflammatory response is protective, but if the inflammatory condition persists for a long time, it is harmful to brain health. Microglia becomes more reactive as it ages, increasing inflammatory response in the brain and suppressing the production of useful anti-inflammatory molecules. Most studies suggest that excessive microglia activity also contributes to cognitive impairment. This chapter tells us how your brain ages healthily. Among them, it emphasizes diet and exercise. Look at the picture. Doesn't it look very healthy? Diet rich in vegetables, fruit, and whole grains, and low in meat and dairy products can reduce cardiovascular risk factors linked to cognitive impairments such as high blood pressure and high level of LDL cholesterol. Numerous studies have found that people who engage in regular physical activity show improved learning, improved memory, and a reduced risk of developing dementia. Physical activity might even slow the progression of Alzheimer's disease and dementia, and higher levels of physical activities have been linked to improvements in some markers of structural brain health, such as reduced skull shortening and less shrinkage in hippocampus. Exercise can also improve blood flow and increase production of neurotrophic factors that support new neurons and synapses. Why don't you exercise even if it's annoying for your brain health? Do you like playing musical instruments or reading? The results of the study say people who perform cognitively demanding work or engage in stimulating activities have lower rates of cognitive decline with aging. Research on the aging brain continues, but there are many questions that have not yet been solved. While solving these questions, we can find better ways to protect the brain. Thank you for listening to our presentation.